welcome to this Nipoppy tutorial. In this tutorial, we will cover how to run processing pipelines on our bits data with Nipoppy. More concretely, we will check the prerequisites for running mriqc on our example dataset, execute the Nipoppy process command, and explore the pipeline output. To participate in this tutorial in a hands-on fashion, you will need a Nipoppy dataset created with Nipoppy in it, data in the bits format in the bits directory of your studied dataset, an installed pipeline setup of mriqc, as well as the manifest file and global config file customized for our local setup and study cohort. If you are missing any of these prerequisites, please check out our previous videos. If you did not go through the previous tutorials but want to start here, we provide you with a dataset in bits format. For this, please download the tutorial dataset from GitHub and then run the Nipoppy init command with the bits source flag, pointing to the bits folder of the tutorial dataset you downloaded from GitHub. Also, please watch the Nipoppy pipeline video since we installed the pipeline setup of mriqc in that tutorial, which is a prerequisite to follow along now. Before we look at the Nipoppy process command, let us do a quick recap of what we did in the previous tutorials, because those steps are necessary in order to successfully run Nipoppy process. We modified our manifest TSV to hold the correct information about our participants, visits, sessions, and available imaging data. We also made sure the right call command for the containers is configured, namely Apptainer or Singularity, according to our available installation. We updated the path to our container store in the global config file. This is either a directory in our Nipoppy dataset or points to another directory like a shared container store within your lab. We converted our raw imaging data to the bit standard, and therefore all our participants have the curation status in bits, as shown by the Nipoppy status command. If this is not the case, please run the Nipoppy track curation regenerate command. And we installed a version of the mriqc pipeline. Additionally, for mriqc, we need the template flow library available. Template flow is a library of neuroimaging templates used by several popular imaging processing pipelines. The first time mriqc is run, it will attempt to download templates to a directory specified in the global config file. We can either create a dedicated directory inside our Nipoppy dataset, or we can point to a more central path to share the templates among a bigger user group. Please be aware that this step requires an active internet connection, as it involves a download action. If you are working on an HPC node without internet connection, please consult the fMRI Prep help page for further instructions. To run the Nipoppy process command, we are specifying the dataset as well as the pipeline name and version. Also, we will first run the pipeline on a single participant in order to see if it runs smoothly or if any errors occur. So we are also specifying the participant and session ID. When we run the command, it will take some time until the pipeline finishes. If we see the message "ran for 1 out of 1 participants or sessions," everything worked well. Outputs of the pipelines are stored in the derivatives directory, and log files can be found inside the logs directory. When we run Nipoppy status, we see that the status hasn't changed yet. This is because the data processing status is tracked by the processing status file, which is created by running the Nipoppy track command. We will explore this command in our next tutorial. Are you ready to run mriqc on another participant now? Congratulations! You are now ready to track the processing status of your dataset.